Tonight's guest speaker, Dr. Chelsea Bennice, is an affiliate assistant scientist at Florida Atlantic University, where she serves as assistant director of community engagement and programming at FAU's Stiles-Nicholson Brain Institute. Dr. Bennice is a lead scientific diver at FAU, and she's also the lead scientific editor for OctoNation, a nonprofit focused on inspiring conservation through octopus education. Dr. Bennice holds a PhD from FAU and has spent more than 1,000 hours underwater studying the octopuses of South Florida, earning her the nickname Octo Girl. So tonight, Dr. Bennice will be telling us more about her exciting octopus-related research. It is with a tremendous amount of excitement that I welcome to the stage Dr. Chelsea Bennice.
All right, thank you, Zach, and thank you everyone for joining me tonight. I'm very excited to be here to talk about my research that takes place in South Florida, talking all about octopus behavior to bacteria. And so I know maybe you can't tell, but I'm in my office, I'm not diving, and I'm sure you guys are in your living rooms or somewhere comfy, but I bet all of us want to be underwater. So I'm going to actually take you on a journey to my study site at Blue Heron Bridge, which is located in the Lake Worth Lagoon. Just like today, beautiful day in South Florida, crystal clear water, and we actually see our first octopus. So we have an octopus right here. This is the Atlantic longarm octopus cruising during the daytime. And shortly, we're just gonna speed up, and it's nighttime, we're on a night dive, and this is the common octopus foraging at night. And so a little more information about these two species. Like I mentioned, the first species that you saw in the video is the Atlantic longarm octopus, known as Macrotritopus defilippi. And so this is a relatively small octopus species. And one way octopus scientists identify or measure an octopus and how big it is they use what's called the mantle length. And that's shown right here by this bracket, kind of termed ML for mantle length. So just to give you an idea of how big this animal is, at an adult size, its mantle length is about 2 to 3 inches. So sometimes I use my pointer finger to kind of figure out like, wow, the mantle length is only that long. That's a really small octopus. Where it gets its common name, longarm, is because its arms are 6 to 8 times the length of its mantle. As you can see here, it's doing this really tall stance, what we call the tripod stance, standing up nice and tall on its arms. And when it needs to swim, it actually wraps its arms on each side of its body and kind of looks like a flounder. And so we'll talk about flounder mimicry a little bit later. So that's a little information about the Atlantic longarm octopus. The second octopus that you saw in the video is the common octopus, or Octopus vulgaris. And this one does look different, but sometimes can be confusing when it's a juvenile at Blue Heron Bridge. And so as you can see right here, for this animal, its mantle length, again, the mantle right here, at Blue Heron Bridge, we see usually juvenile or subadult octopuses there. The mantle length is between 8 to 10 inches. So it can get a lot bigger than that Atlantic longarm octopus. And its arms, you can see they're a little more robust or a little bit thicker than the Atlantic long arms, and they're shorter. So they're about 3 to 5 times its mantle length. And just for another size comparison, the Atlantic long arm, it weighs about a pound or less, and the common octopus weighs a few pounds, but it actually can weigh up to 11 pounds. So it can be a relatively larger species, especially at an adult stage when we usually find it out on the coral reefs. And so the big question, as you probably saw from the video, is that these two species coexist at Blue Heron Bridge in the Lake Worth Lagoon and also other areas. And the big question is, I see a lot of these two species there. How do these two species coexist without competing for resources? And so that's a big research question. And to answer this big research question of coexistence, I want to break this down into specific research questions, figuring out what types of resources each species use, and if they're partitioning or using different resources to coexist in this lagoon. So the first research question is, where exactly do they live at Blue Heron Bridge? Are they spatially partitioned? Do they live in different areas or not? Are they there year round? Maybe they're showing up at different times of the year, so they're not running into each other. They don't see each other that much. What do their homes look like? So the homes of an octopus, you'll hear me use the term den. So if the octopus's den is made out of rock rubble structure, when are they active? So when are they going out to forage and feed? Are they going out at different times of the day to avoid each other or not? And lastly, what do they eat and how do they eat? So looking at their diets and also their foraging tactics of where they get their food and how they get their food. And so this study takes place, all of it at Blue Heron Bridge, Phil Foster Park. I like to call myself a field biologist. I'm the happiest when I'm underwater. So this was a really fun long-term project that I was able to do. So what I would do, Blue Heron Bridge is easy, accessible. It's a shallow water location, so it allows me long dives, about 2 to 3 hours, so I can get a lot of data, or I can study all this information about the different octopus species. So I park inside the park, I gear up. As you can see, I've got all my science gear in that top photo, and then it's just a beach dive. I walk directly in from the beach to the shallow waters. Basically what I do is I swim north and south paths looking for octopuses in their dens. Once I find an octopus, since this is a shallow dive site, the average depth about 10 feet, I'm safely able to ascend to my dive float where I have a Garmin GPS device. And I'm just going to circle it with my mouse right there. That's a Garmin GPS device that I keep in my dive float. So when I ascend up, I mark the location that I found that octopus, and then I descend back down, and then I just continue swimming up and down Blue Heron Bridge, recording all the octopuses. So I did this year round for 3 years, and showing up are green GPS locations for the common octopus, and those pink locations mark the dens of the Atlantic longarm octopus. And to take a snapshot of that over Blue Heron Bridge, this map you're going to become very familiar with, you see in green the common octopus, and then in that pink you see the Atlantic longarm octopus. And you can see that there is a lot of overlap. So again, a lot of time underwater, 3 years, 371 dive hours, and you can see that both species occupy the west side of the Lake Worth Lagoon, Blue Heron Bridge, and also the east side right here. There are some dispersed throughout the sandy area. However, most are clustered on the west and east end, showing that there is spatial overlap between the two species where their occupied dens are located. So we know that spatially, they're underwater neighbors. They're living right next to each other. And so the next question was, okay, well, they're there right next to each other, but what does the data look like when we separate the data out into months? So categorizing into seasons. So I took those 3 years of data information, I separated into winter, spring, summer, and fall, and looked at the number of octopuses or octopus abundance and how that changed for each species over those categories. And I saw that both species, they have the same seasonal trend. As you can see, in dotted line is the common octopus, solid line is the Atlantic longarm octopus. Not too many octopuses there in the wintertime. You see both of them jump up, increase in that number of octopuses during the springtime. There's still quite a few there during the summertime, and then it drops off And sometimes on research dives, I would see 0 octopuses during the fall, And so that's what I've termed now octopus season. It is March. So if you want to see an octopus, now's the time to go diving at Blue Heron Bridge. And to just give you an idea of how many octopuses I've counted on a single dive, sometimes I'll count up to 10 octopuses on one dive, and that can be for one species. And so they're definitely there, they're abundant, and a lot of them are juveniles that are at Blue Heron Bridge. So that answers part of the big overarching question of how these two species are there or when they're there. So just a quick recap, we know that they're there the same time of year. They are living right next to each other, underwater neighbors. Okay, so how are they doing this and not competing for resources? So then looking into their habitats, they live right next to each other, but maybe they're using different habitats or microhabitats. So next up on my research dives, after I would take that GPS location to send back underwater, I would then take a picture of the octopus's den. And so I would term this a photo quadrat. So you can see right here, this is a PVC pipe in a square, what we call a quadrat. It's a known area that I'm taking a picture over. So if I were to take that picture directly over it, this is what I'm taking a picture of. There's this tiny little octopus inside the middle of the quadrat, and I term that the den habitat. Next, what I do is I move that quadrat around that middle square and take more photos, and I consider that the surrounding habitat. Then I take all of these photos and I put them into a software that helps me calculate the percentage of substrate composition for each octopus. So I did this 20 times for the common octopus. I did it 20 times for the Atlantic longarm octopus. And then I just wanted a general idea of what the habitat or what the substrate was at Blue Heron Bridge. So I did 20 random spots at Blue Heron Bridge to get an idea of the general makeup of Blue Heron. And so after 810 photos, or I did, it would be 40,500 points because I did 50 points per photo. So this is the not so fun part of the research, but you get really cool results after taking all these pictures and analyzing them with all these points. We see that the two octopuses have different habitats. So they have different immediate habitats that I term the den habitat, and they also have different surrounding habitats. The common octopus is found around hard bottom, so it's found around rock, shells, and rubble. But the Atlantic longarm octopus is found on soft bottom or sand. So the common octopus needs structure or something to create its den out of, where the Atlantic longarm octopus just has this little, it's probably about the size of a quarter. About the size of a quarter opening, and what it can do is it uses its really long arms to create that hole or that burrow that it then lives in. So it's a sand-dwelling octopus as opposed to the structure that the common octopus needs. So we see that since the Blue Heron Bridge has a patchy environment, and that's what I found out from doing the random locations, analyzing those. Since it has a patchy environment, these two species can be underwater neighbors and coexist. Remember, if you go back to that first map, I had all the green and magenta dots. We saw that both species were over in this location on the west, and they were also over here on the east side of Blue Heron Bridge. However, patchy structure, this is just a general representation of rock, rubble, and structure by these circles, and then sand represented by the orange circles on both sides. Since we have this patchy environment, allows these two species to coexist right next to each other. So that answers part of the story. However, usually species use multiple mechanisms of coexistence to varying degrees. So I definitely wanted to look at other resource partitioning mechanisms. So this one is looking at their activity times. And to look at activity times, you need to know what the animal is doing 24/7 or 24 hours out of the day. And I don't know about you, but I like to sleep. And so usually my research dives, they would take place from 6 a.m. in the morning until the park closed. And I would do night dives till 10 p.m. But that leaves a lot of time where I don't know when the octopus is diving. I really don't want to do a dive at 2 a.m. or 3 a.m. in the morning. So we came up with a 24-hour camera called the Octopus Monitoring Gadget, or the OMG. And what this is it's a GoPro camera inside this underwater housing. We used a PVC pipe. We have an external battery to keep the GoPro running longer than the 2 hours, so getting a full 24 hours. A very large memory card for all this information. And of course, we used a red LED light for nighttime footage. Octopuses are insensitive to the red wavelength, so we wanted to make sure we did not disturb or alter their natural behavior. So a red light was used for nighttime footage. And so this is actually the first video of deploying the Octopus Monitoring Gadget. Here's my dive buddy, and there's a pipe right there, and there's an octopus living inside the pipe. I'm the one carrying the OMG. Fast forward, it worked. The camera didn't flood or anything, so all was great. And you can see the octopus comes out, and what I'm looking for to consider activity time and foraging is I look for the octopus spreading its arms across the substrate. I call it speculative foraging behavior, and also bringing that arm kind of to the middle of its body. That's where its mouth is located. And so I know, okay, this animal is foraging. And I'll mark that the animal was out during this time to forage. You can see that this octopus goes back for a short amount of time. Fast forward, leaves again shortly after midnight. I won't make you watch hours of video. And then the octopus does a grand entrance back in. And so that octopus left shortly after midnight. I think that was like a 90-minute foraging event. And so it was nice. I was actually at home sleeping when this octopus was foraging. And so I deployed this several times for each species. As you can see here, I reviewed over 1,000 hours of octopus video, and I got some really great data from that. And this was actually the first study that has looked at octopus activity times for a 24-hour period under natural conditions in the field. So it was really exciting to get this information. And this is actually the first study to look at activity periods for the Atlantic longarm octopus. And so what you can see here is dotted line is the common octopus, solid line is the longarm octopus. They're complete opposites. You can see that the Atlantic longarm is coming out during the daytime, peak foraging around 1 p.m. And then you have the common octopus at nighttime. Foraging usually around, it looks about 10 p.m. is about its peak foraging time. And so we have that they live right next to each other. They use different substrates. They're coming out at different times of the day to avoid that interference competition or running into each other. But they could still be eating the same thing, just coming out at different times of the day. So the next thing is to look at what they're eating. What is their prey? And so a common method to look at the diet of the octopus is to collect their prey remains. They're usually eating some prey that has a hard external structure. And so you can see me, here I am lining up the prey remains of this octopus right here. This octopus isn't a big fan of me moving its prey. Usually they'll use the shells to kind of barricade or put around their dens to make the opening smaller so predators can't get inside. Octopus said, if you're gonna check out my den, well, I'm gonna check out your camera. So you can kind of see here, here's me scuba diving with my underwater camera in the size of this animal, subadult juvenile animal, mantle length, probably, I'd say about 6 inches, not too big. So what I'll do is I'll snap some photos of the shells, some of them that cannot be used as a home by another animal, I will take with me, and I will ID 2 species if possible back at the lab. So that's one common method used, is collecting the prey remains. However, I found out that not all octopuses leave prey remains around their den. And of course, this was the case for the Atlantic longarm octopus. Since it lives in sand, if it were to leave prey remains around its den, it may actually attract attention to itself and doesn't want to attract any predators. Octopuses have a lot of predators. So I would rarely find prey remains left around their den or their hole opening. And so what I had to do, or what I did to get a better idea of what the diet was for these 2 species, I would follow them on foraging events. And so I would follow the octopus along foraging events, record the foraging behaviors, and if I could actually capture them, in the middle of catching prey, as you'll see right here, this crab jumps out. And so this octopus gets it. So that is one data point with that information. And the octopus isn't done eating. It actually continues on and looks for more prey. As you can see here, reaches his arms out. There's another crab in that sand. The octopus definitely senses that with its arms, and it has 2 yummy meals. And so you can see here, it's eating on the run or the swim. And so this is information that I would have not gotten if I would just look at the homes or the dens of the octopuses. Also, the carapace of the crabs that they're eating, that's crunched up, they usually spit it out and the current takes it away. I wouldn't be able to have any of that information. And so by using multiple methods, I was able to get a better idea or survey of what these 2 species were eating, if their diets overlapped or not. And so what we saw here is that I broke up the prey into 3 categories: bivalve, gastropod, and crustacean. And we see that the common octopus eats mostly bivalves, then gastropods, and then crustaceans. Well, it's reversed for the Atlantic longarm octopus, like you saw in the video, where it just snatched up 2 crabs right away. And you can also see representation of that in these 2 photos. So right here we have the common octopus, and the most commonly consumed bivalve was actually this one shown right here, species named Chione elevata. And one of the main crabs that the Atlantic longarm consumed was this shamefaced crab, or also a box crab. So a box crab was definitely a favorite. And just a fun fact as well, that the 2 species also consumed mantis shrimp. And so mantis shrimp are usually considered some pretty tough animals, but the octopus was able to take it. They were the smaller false mantis shrimps, but still, that was a pretty cool interaction that I was able to see. Another cool thing about looking at octopuses and their diets and octopus behavior is that I mentioned before that they are sensing the environment. It's kind of like a sensory overload. They're benthic animals, so they're taking in their surrounding environment and looking for prey, but also making sure they're not detected from predators. And to do this, they use multiple sensory organs or cells. And so vision, one of them being that you've probably noticed that the octopus has a really weird looking pupil. Ours is circular, theirs is usually rectangular unless it's dilated. So this rectangular pupil actually allows them panoramic vision. So they have that rectangular pupil, but also their eyes sit on the side of their head. So it allows them almost like a 360 view of their environment for predators and prey. They also smell the environment. So it would look pretty weird if an octopus had a nose. So instead, they have sensory cells or receptors in dimples that are all over their skin, mostly their mantle, and it's smelling chemicals, distant chemicals in the water to see what that is, maybe a prey source. And of course, their arms and their suckers. And so octopuses have lots of sensory cells along their suckers. And to just give you an idea of this, so you have one sucker right here. Along the rim of the sucker, there's 10,000 sensory cells called chemotactile receptors. So that means that the suckers can taste and touch what they're coming in contact with along the bottom. So they can decipher and figure out if it is a crab, if it is a rock, if it's something they want to eat or not, and the shape of it and what it tastes like. So 10,000 sensory cells on each sucker. They have 200 suckers on each arm. Octopuses have 8 arms. So that's 16 million chemotactile receptors. So when I say sensory overload, they're taking in all of this information and then processing it to figure out their surroundings. And to just give you an idea of how many taste cells that we have on our tongue or taste receptors, we have about 50,000. So that is a lot of tasting and touching that the octopus is doing, but it needs to, since it is a benthic animal. And so a little bit more about after it catches its food, after smelling, looking around in the environment, touching and tasting with its arms are super strong. And as you saw, I'm going to flip back really quick. Look at the size of this is an upside down crab. And so you can see that this octopus is able to catch and eat a prey item that can be its size or much larger. And that's because the octopus is super strong. It can hold on to that animal, but also it can start breaking down that animal before it actually consumes it. And there's different ways, or there's multiple ways to do this. So as you can see here, we call this the arm crown, where all of the arms meet together. And in the middle of that arm crown is actually the octopus's mouth. And you can kind of see over here, better shown in the diagram. So what the octopus can do to break up that exoskeleton to get to the yummy stuff or to break apart that bivalve shell is it can use its hard beak, very similar to a bird beak, as you can see right here, to crack it open. However, if the shell is too thick or the octopus can't open the shell, or if the animal is moving around, or maybe it's a crab and it's about to pinch the octopus, what the octopus can do is it actually has a tiny little drill in its mouth called the salivary papilla right here. And that's connected to its salivary gland. And so the salivary gland produces the saliva, but also in the saliva is a cocktail of toxins and enzymes. So what the octopus does is it drills that hole into the shell or an exoskeleton and delivers that saliva. What that does is it actually paralyzes the animal and it starts to break that animal down. And so just to show you an example of this right here, so you have it scooping up that bivalve, and if this octopus can't open that bivalve shell, it will drill a hole. It will inject that saliva. One of the main toxins in the saliva is called cephalotoxin. And so after that, the animal can't move. The octopus starts to break it down using different enzymes. And it's almost like I like to call it like a crab or bivalve soup. So the octopus's brain is actually a donut-shaped brain. And so it actually goes around the esophagus. So the octopus can't eat or swallow large pieces of food, or it may poke or injure its brain. So it needs to break its food down before it brings in that food to the esophagus and then to its digestive system. And so what I also did is I looked at if octopuses use the drilling technique and if they use them for a certain thickness of shell, but also maybe if they used it if the animal had an operculum, like a conch, like a trap door or not. So as you can see here, measured the different thicknesses of the bivalve shells and saw that there was no drill hole for shells that were 0.4 millimeters thickness. However, if you almost double that thickness, you see that there are drill holes that the octopus uses when it is a thicker shell that maybe it can't pull open. And so that's a circle right here for the side of the drill hole. And if you see this arrow right here, it points to that drill hole is actually in a strategic place where the bivalve's muscles attach. So it injects that toxin straight into that muscle, kind of shown here by a darker gray circle, to make sure it delivers it efficiently and paralyzes that bivalve's muscle. And so for gastropods, looked at if they have an operculum, that trapdoor or not. There was no drill hole for gastropods that did not have an operculum, and there was a drill hole, as you can see here with this conch, if they did have that trapdoor. Again, that drill hole was at the site of muscle attachment. And also, I don't have it displayed here, but for a couple of the crabs that I was able to sample and keep, I looked at the crab claws, and you can see there are drill holes in the crab's claw where the muscle is, where they drilled a hole in the crab claws. And so we have here is that the two species are living next to each other, coming out different times of the day. There's some overlap, but they're eating different things, and there are different strategies to consume these items. And this leads into where they're eating and how they're eating these items. And so this is looking at the foraging strategies, the tactics of each species. So not only did I follow the octopus to see what it was eating, I also followed it to record its foraging behaviors and the different types of substrates that the animal was foraging on. So to give you an example of this animal is using what we call grope searching, where it takes its arm, goes down into a crevice, searches for an item, and it's on hard substrate or rock. So you can see it moves. It's not actively doing anything, so it's sitting on soft bottom or sand. And then it moves over, and that's called a parachute attack, where that arm webbing comes out, traps any prey underneath on soft bottom. And another parachute attack, however, on this time, it's on hard bottom. And so those were some examples for the common octopus, and a few other examples for the Atlantic longarm octopus. Again, there's this tripod stance that this animal does. I think it's like a lookout tower or like a lifeguard tower, kind of surveying, scanning its environment, making sure no predators are around. And then it goes off and does this flounder swimming, mimicking the flatfish or the flounder. Speculative bottom searching, where it's just taking all its arms out, searching around. Maybe that's another mimic octopus. Again, speculative bottom searching, just reaching his arms out. Once that speculative bottom searching, that arm goes down into the hole, it's considered a grope searching, kind of checking out if there's maybe any crabs down in that hole. And so I showed you a few behaviors. I used several behaviors. However, let's first talk about the substrates. This kind of is set up the same way as looking at their substrates that their den was located in. So looking at fauna and flora, so that's like seagrass, sponge, could be coral, hard bottom, that is rock rubble, shells, soft bottom is sand, and human debris can be, sometimes the octopuses do live in glass bottles or cans or cement blocks at Blue Heron Bridge. And you can see here, just like their dens or their habitats, they have different foraging substrates. The common octopus foraging mostly on hard bottom, but you can see kind of it's foraging across all the different categories. And you can see for the Atlantic longarm octopus, 3 of the 4 categories. However, it's really dominated by the soft bottom. So thinking of the Atlantic longarm octopus being a sand-dwelling animal, a specialist eating crabs, versus the common octopus being more of a generalist, going over all these different types of habitats and also eating the different types of prey items. Next up, we have these different types of foraging behaviors along the bottom of the graph right here. And so these were the most popular behaviors used, and they were used about more than 80% of the time the octopus was foraging, and then the percentage of time that they used them along the y-axis right here. So both animals commonly use speculative bottom search, and you can see right over here on the left side of the screen. And this is because this is more of an exploratory behavior that both species do to first check out the environment and what may be there. Then, depending on where they're at, they may use more specialized behaviors or species-specific behaviors, as you can see by the stars. So the common octopus using the parachute attack, like you saw in the video, and then the Atlantic longarm using that flounder swimming mimicry, and also that tripod stance. So they have some of the general behaviors that both of them use. However, they have species-specific behaviors to catch the different types of items on the different types of substrate that they are on. And just one more thing that I wanted to point out about their foraging behaviors and the foraging tactics is that there's a lot of stationary or pause behaviors. And to think that I think it's more than 30% of their foraging behaviors were pause behaviors. And I like to tell everyone that octopuses aren't lazy just because they're sitting or standing. They're actually, this is the time that they are processing, that they are scanning the environment, taking in this information, and then learning and building those memories. So it's very important for them because they are a soft-bodied animal with a lot of predators, and they have to make sure that they optimize their foraging by taking this time, pausing, finding an area where there are plenty of prey or resources. So you can see that there is a lot of sitting involved and a lot of tripod stance for the Atlantic longarm. But this stop and go or saltatory search behavior is very important for this animal and how it plays into how they forage and feed in these different environments. And so just to give you a big recap of this big 3-year project, looking at how octopuses use different resources to facilitate coexistence, we have that they're underwater neighbors, their diets, there's some overlap, but we see that the common octopus feeds mostly on bivalves, the Atlantic longarm mostly on crustaceans, those crabs. They live in different homes, they come out at different times of the day. That allows them to coexist by using multiple resource partitioning mechanisms at the study site. And so it's really important to understand the resources that these two species need to coexist, because it increases biodiversity and because octopuses play important roles in many marine food webs. So we need environments, and we need to know the specific environments that octopuses need to have enough food for when they're grazing on lower trophic levels. But also, like I've mentioned, they have a lot of predators. So a lot of those apex predators need a food source for them to consume. And so speaking of predators, not only did the octopus monitoring gadget give me a lot of information about their activity times and the prey that they're eating, but it also gave me information about what else is going on in their environment. And so octopuses have lots of predators. This one is from above. This is a diving bird, a diving cormorant. You can see here that wants a piece of this octopus. They're high in protein. They're a squishy protein treat. So every predator is trying to consume them. And so another thing that I like to point out is that not only is another octopus being there of a different species maybe altering when they come out to forage and feed, but this area, if there is a high density in predators, it may be altering when the octopus is coming out. So it's important to always think about the different components of the environment and what is altering, or maybe what the octopus is adapting to allow it to coexist there. And so you saw that came by these different predators. So we had the bird from above, we had the fish in the water column, and then we have this benthic predator, this stingray. Now you can see here, this is at night, that red light from the OMG footage. The octopus leaves, we speed up, we have the purple mouth eel. And so eels are known to specialize on octopuses. And so if we fast forward a little bit, you can see here also the eel took over the octopus's den, is waiting for it. You see that the octopus is waiting, slowly creeping back. Might be hard to see, so I'll put my mouse over it. You see this eel is waiting until this octopus comes back, thinking that it's got a nice meal. Wait for the suspense. You can see that octopus did not use any slow crawling behavior. It used its fast getaway, its jet propulsion to get away from that eel, so it was not dinner. So the octopuses face predators, and you can also see here that during the springtime, I see increased fights of octopus. So there are an abundance of octopus during the springtime. I see fights, I see den takeovers here. I see octopuses kick each other out of the dens. And so octopuses may use other methods, to coexist than the ones that I discussed earlier. And so just to fast forward here. And this is just to show you an example. Here we have an Atlantic longarm standing nice and tall. Here's the common octopus right here. And what they'll do is they'll extend their arms. Remember, chemotactile cues on their suckers. What I like to call the octopus handshake. They can visually see that another octopus is there. However, they don't know if it's the same or different species or different sex. So they'll do the octopus handshake, loaded with the chemotactile cues on their suckers, do that handshake, and they just saw that, like, nope, this is not the same. This is not the same species. I don't want anything to do with this animal. So like you saw in this video, living really close to each other, these two were actually both out during the daytime. So another mechanism or method to avoid each other is this communication at heightened times when there is an increase in abundance of these two animals. So just more things to think about that octopuses have, evolved and they adapt and they use to allow them to coexist, multiple species to coexist in one area. And so it was really cool to gain all of this information of these two species at Blue Heron Bridge. It kind of served as like the baseline or the backbone for other research projects that have just finished up or that are going on right now. And so few projects that I've just finished up or that are going on right now have to do with species identification, octopus arm flexibility, and also the octopus microbiome, what bacteria are living on their skin. And so just to bring our friend back, the common octopus, you can see here that a lot of the time, octopuses look the same. And of course, they can change color. So then it just gets really confusing trying to ID these animals if they have similar colors, color patterns, and they also can change them. So who's who? However, they do have species-specific patterns that help scientists identify the different species. So we don't have to take tissue samples to identify the species. And so just to give you an examples of this, we have the common octopus. It's common that has not only do they have different color patterns, but also their skin texture. So I haven't really talked about that, but just a quick overview. Not only can they change the color of their skin, they can also raise their skin to form different skin textures. So they can be smooth, or they can be bumpy, or in this one, they can have spikes. And so usually for the common octopus, you can have 4 large spikes along its back. They're called skin papillae. That's common for this species. It's hard to see in this one, but they have 2 white dots in front of their face. And also this mottle coloration is common as well. Also underneath their suckers, the ventral or the underside of their arms can be a pale orange to a dark orange to a white. So it's usually a solid uniform color. Now, this animal would look very similar to the common octopus. However, this animal is actually showing a dematic display or a threat or startled display. So usually this area, just like you saw in the common octopus, looks very, very similar. And these species, this photo was also taken at Blue Heron, so these species overlap as well. However, when they do this dematic display or this threat startle display, it's unique and it's only specific to the brown striped octopus. And why it's called that is you can see it has these longitudinal brown stripes that go down each arm, and also this very distinct, what we call an eye bar right here. So that identifies that species. Next, we look at the Caribbean two-spot octopus. And so again, a lot of octopuses have this orange, brown to tan coloration, also depending on where they live. However, this is a relatively small octopus, probably around the size of the Atlantic longarm. The difference between this octopus and others, like its name says, is the Caribbean two-spot octopus. So it has an eye spot, a false eye spot, usually around its second to third arm right here. And I'll put a circle around that. It has like a blue iridescent ring. So this is not to be confused with the blue ringed octopus. We do not have that octopus here. It's an Indo-Pacific species and they have 50 to 60 rings. This octopus only has 2 spots. So if you see it diving, it's not a blue ringed octopus. And just another example of this, again, a very small octopus. And again, one eye spot, false eye spot on each side, just below its eye. And so this brings me to the latest research that I've done, that I was a co-author on, that we published, is looking at this Brazil reef octopus species. And a really cool aspect of that is citizen science. And so you can see in the top picture, this is called Octopus insularis, or the Brazil reef octopus. And you can see it looks very similar to the common octopus, or another species called Octopus americanus. However, the main visual distinguishing feature, one of them being the underside of its arms, or the ventral arms. So again, this animal, Octopus americanus, orange. The Brazil reef octopus, this kind of, we call it reticulate pattern, or kind of a speckled white-red pattern. And so I was contacted in 2018 by a scientist in Brazil. She read the paper on the 2 species of octopus, Octopus vulgaris and the Atlantic longarm, coexisting. And she asked me if I had ever seen this Octopus insularis in Florida. And I said, no, I've never seen that. And she said, are you sure? And I was like, yeah, I'm pretty sure. I'm pretty sure I know the different species that I'm looking at. And she said, look for the red and white arms. And I said, okay. I looked through my photos and I hadn't seen any. And so they were interested, and she and another co-author were interested in coming up with a field guide, because these 2 species get confused a lot. And so we did a call to collect lots of photos from underwater photographers, and then of course our photos. And so these photos started coming in from Florida. So I was doing all the Florida photos, looking through them, if they were Octopus americanus or vulgaris, or if they were a different octopus species. And so I start looking through them, and I noticed one from West Palm Beach. I said, that's not Octopus vulgaris. That's not the common octopus. And then another one comes in a little bit later from Lantana, and I was like, that's not the common. This is very recent. This was just about a year ago. We published this in 2021. And then another one from Boynton Beach. And I said, well, insularis is here. Welcome to Florida. So this Brazil reef octopus is now in Florida. We hadn't seen it before, but we're starting to see it now. We just wrote up a field guide for everyone to be familiar, so we can help, with this identification so people don't misidentify it. And I just heard back from a scientist that's doing the genetics from a Florida specimen of Octopus insularis. And so we do have confirmation, just not visual confirmation that it's insularis, but genetic confirmation that insularis is in the Florida Keys and likely in South Florida. And so just to give you an idea again of when I saw this on Lantana, my friend actually took this picture and she contacted me and said, what species is this? And I said, I think this is a new species. Take me to where you found this octopus. And so we went snorkeling off of, the Lantana beach. And just in that snorkeling event, I counted three Octopus insularis. So they are definitely here. And we think that they're here. This is a tropical octopus. It likes warm water. It was thought before that Florida was too cold. I know that's hard to believe, but Florida is technically subtropical. And so they thought it was too cold. However, with warming water temperatures, this octopus could be moving further north. And so we proposed, a geographical expansion of its range further north, possibly due to, ocean warming events. However, this is another example of it possibly using different habitats than Octopus vulgaris and other animals. So we may have multiple species coexisting in our Florida coastline. I haven't seen this at Blue Heron yet, but that doesn't mean it's there. Hopefully I'll be in the water soon, and stay tuned for that information. And so just wrapped up that species identification project. However, I'll be doing more work with a master's student to look at cryptic species of octopus to try to figure out who's who in South Florida and throughout the Caribbean. This then leads me into the second project that's ongoing now, and it's in collaboration with Dr. Roger Hanlon's lab up at the Marine Biological Laboratory in Woods Hole, Massachusetts. And so since I collected all of that field foraging behavior video, I was able to use it for this project, looking at octopus arm flexibility. So octopuses have no rigid skeleton. They are made up of muscular hydrostats. So you can think of a muscular hydrostat like our tongue or an elephant's trunk. They have about 4 different types of muscles that make up their arms. And basically this allows them an infinite number of degrees of freedom. So this basically allows the octopus to bend, twist, and move its arm in many different ways, which is definitely bio-inspiration when you're looking at soft robotics. So we wanted to look at how octopuses use their arms, how they're flexible, and apply that to the soft robotics field. So first, before doing all of that, we have to figure out how you're going to analyze and score octopus arm flexibility. And so when you're looking at a video, you have to make sure that you look at the same arm, the same time all the way throughout the video. So we name the arms. And so you have octopus arms are referred to in pairs. So you have left and right 1, left and right 2, left and right 3, and left and right 4. And all looks good here because the octopus is in a straight line, the arms look good. But when you get to a picture, it's never that perfect in a diagram. Their arms are going everywhere. And this is just a quick snapshot of what the octopus looks like in the field. And so what we have here, it's always the octopus's right and the octopus's left. So just to take a quick snapshot of it in the video, right and left 1, right and left 2, right and left 3, and right and left 4. And so what we're doing is we're taking a hierarchical approach. And this is just to give you an example right here. This is one of my foraging behavior videos. So we have these complex behaviors like I demonstrated before, like parachute, or in this example, walk. So we have 15 behaviors. What we did is we took those 15 behaviors and we broke them down into simpler arm actions. So what arm actions make up that behavior? Is it a reach? Is it curl? Is it trailing? So there's a few examples of that. Next, what we did is then we broke those actions up into the 4 deformations. And that's really getting into the flexibility of the arms and building maybe that soft robotic. And so those 4 deformations, we're looking at the bending, elongating, shortening, twisting, or torsion of the octopus arm. So just like a quick example, in here we have the octopus is walking. You can see it's walking on arms. And so just focusing on this arm facing us right here, we're going to highlight that. And the one action that it's doing multiple actions at one time, but the one action that it's doing is reach. And then within that reach action, one bend that's pointed out is right here, where you can also see that the arm is bending down here and bending right here. So just imagine that this is one arm. We have to do this for all 8 arms for a minute of video and do this for lots of videos. And so right now we've analyzed almost 4,000 arm actions. And what we've seen so far, that not to our surprise or anything, that these arm actions are used across multiple behaviors. You can see octopuses are multitaskers. They've got a lot of space, a lot of substrate to cover in a quick amount of time. And they have to analyze what's going on in their surroundings. But what's cool is that we did see that some of these arm actions are specialized for specific behaviors, like walking, and that they do partitioning between their anterior and posterior arms. So anterior arms being the 1st and 2nd arm pair, and the posterior arms being the 3rd and 4th arm pair. And so a lot of what we see for this partitioning, and walk is a good example, that they only use their 3rd and 4th arm pair for that reaching or that standing. Usually their front arms are curled back. And so knowing what specific arms are doing, the deformations that we use or that they use, we can apply that to soft robotics and the engineering community. And so stay tuned later on for that. We're almost done grading the deformations, so that will give us more insight into how the octopuses are using their arms. And so last but not least, I'm also looking at the octopus bacteria, so their microbiome, what grows on their skin. And this is in collaboration with Dr. Jose Lopez's lab at Nova Southeastern University. It's been known that bacteria can play a key role in cephalopod lifestyles. Just a few quick examples of this is the bob-tailed squid. They have a light organ that allows them to do a type of camouflage called counterillumination. So they have a light organ in their mantle. Inside their light organ actually lives symbiotic bacteria, bioluminescent bacteria that produces this light. And so it's very crucial for them to have this bacteria, allows them to do this counter shading or counterillumination to hide from predators. Another example I think we're all familiar with is the blue ringed octopus. And so one of the most deadly octopuses having that tetrodotoxin or TTX that's actually in their salivary glands or in their saliva. And so the octopus does not produce this toxin. It's actually another symbiotic bacteria that lives in their salivary gland that produces this TTX. And so what I'm looking at is how bacteria on the skin, if there is any if there's any bacteria on their skin that's unique to the octopus that helps combat them from infection. And so just to stress again how important it is for octopuses to remain in many marine food webs. We need to make sure that they remain healthy. And a big part of this is combating bacterial infection. Bacterial infection is one of the main or leading causes that, our coral reef animals are in decline. And so octopuses right now, they're healthy, they're thriving. We want to see what is helping the octopuses stay healthy. They have a very simple immune system. So we are hypothesizing that they have like I showed before, those symbiotic bacteria that were in the other examples, that octopuses also have a symbiotic bacteria that live on their skin called their microbiome. And so to look at this, no one's really has done this, looking at how to swab an octopus and to actually how to swab an octopus for a bacteria sample in the field. And so this is the first study that looks at their microbiome under natural conditions. And so octopus skin has a mucus layer around it. So we're thinking that it has the mucus, the bacteria. If we can swab underwater, it will stick to, it's a swab. It's very similar to a Q-tip. That mucus will stick to the swab long enough to transport into a tube. So we were thinking that, well, we didn't want to remove the octopus. We're trying to make this as, least invasive as possible. And so here I have, and you can kind of see how small, this is an Atlantic longarm octopus, how small this animal actually is. And so here I am giving it a swab or a little belly rub, what I like to call it. The octopus is just hanging out. And so making sure that I do a good swab on its skin to get mucus and bacteria, and then put it into what's called the transport tube. And so that was the first round of figuring out the methods that did not work out. And so then COVID hit and I had to be creative. I had to think about, I couldn't dive, scientific diving was shut down. So I had to figure out, read some papers, how am I going to swab an octopus, get a better swab sample because I can't swab this animal underwater. And this is when we came up with the ROV Octo, or my floating lab. I knew that I had to take them out of the water. However, I didn't want to collect the animal, have to transport it to somewhere on land, swab it, and then take it back out. So what I did is basically I developed a fancy floating lab or a boogie board. And what I did is I cut out areas to hold my sampling equipment. So it holds my swabs, it holds my sediment samples, it also holds my water samples. And of course, we have an octopus holding bin. And so this is a removable basket, which allows me to take the basket underwater to collect the octopus, to bring them onto the floating lab. And then you can see this, the basket of course has holes in it. So the octopus is actually in the water. So when I'm preparing to swab, the octopus is getting water, the octopus is getting oxygen, and it is fine. And I'm minimizing stress. And so let's just see this in action so you guys can see what I'm talking about. And so here we have an octopus in my octopus basket. And so I put just a lid on. Again, since I'm sampling in the lagoon, it's very shallow and I'm able to do this. I'm ascending up to the floating lab, as you can see up there. And here I am checking to make sure the octopus is in there. The octopus is getting water. And what I do is these are the swab samples right here. They're sterile. I can have to make sure that I'm not touching anything besides the octopus and its mantle, mucus, and bacteria. Once I'm ready, I lift the octopus out of the water. I do a quick swab. Once I'm done, I take the octopus and then I release it back into the water. And then you have it right there. And I release it back where I found the octopus, or in an area where you see their structure. Octopus can go for shelter. After I'm done swabbing the octopus, I then take a sediment sample of where I collected the octopus and also a water sample. So this is to compare the bacteria that I find in the water sample with the bacteria I find in the sediment sample, and then also the bacteria I find swabbing octopus, to see if there's a unique set of bacteria that compose the octopus microbiome. And so all the samples have been collected for that. Hopefully I am sending them off to get sequenced to understand what types of bacteria or bacteria species live on the octopus. And so stay tuned for those few octopus projects that I have going on. I can keep you updated if you follow me on social media. That is Facebook, Octo Girl, or the Instagram, The Octo Girl. And you can stay updated, and hopefully I'll be invited back and I can tell you about the different results from those projects.
Chelsea, we will definitely be inviting you back. That was just an amazing, amazing presentation. It's, even as a scientist, it's hard for me to wrap my head around the amount of work that you've put into these projects. When I'm diving, I love to run into octopuses, but I've never seen the vast majority of these behaviors. And you spend so much time down there that you've become, intimately connected with these animals. And it just blows my mind that you've been able to do all of this right here at a local inshore dive spot. So thank you for coming out tonight. We've got some amazing questions rolling in. Please keep adding to those questions. We've got about 20 minutes or so to go over your questions. I hope I can get to all of them. I'm already looking through them, and you've got some good questions ahead of you. One of the questions that a couple of you have asked, and I'll get to this one right off the bat. Will this lecture be posted online for people to view later? Absolutely. All of our lecture series presentations are recorded and posted to Florida Oceanographic's Coastal Lecture Series website. So for those of you who are joining us for your first time tonight, we have 7 other lectures from this year that you can go back and watch. We also have a bunch from previous years. Lots of really cool content for you to check out. I always say, as long as you pretend it's live, it'll feel like it's live. So be sure to go back and check some of our other presentations. Our first audience question: can an octopus live in an estuary, or do they need the higher salinity of the ocean?
So yes, they can live in an estuary. So Lake Worth Lagoon is actually considered an estuary. So they're able to live, in estuaries. So I would also say, depending on how close that is to an inlet. So Lake Worth Lagoon is very close to the Palm Beach Inlet. It gets that fresh seawater source coming in. So different factors, but yes, they can.
Great. Can your 2 study species interbreed?
Not that I'm aware of. I have not seen any hybrids.
All right. One of your early slides was a map showing the locations of the dens for the 2 different species. And one of our viewers noticed a higher concentration of dens to the right and the left of the island, kind of in the channel areas, with less around the bridge. And I know you talked about substrate being an issue, but they were wondering whether just the man-made island might interfere with water flow and impact den location. Do you have any thoughts on that?
Yeah, that's a great question, and definitely looking at areas of water flow, I feel like it's definitely a future direction for the research. We do see that they're located there. One reason is because of that patchy habitat, but also if water flow is important, if there is more prey resource in those areas because of the different habitats. So there's definitely more information to look at to see why they may be clustering in those 2 areas. Also because of, I guess, the foot traffic or snorkel traffic, possibly in the snorkel area. There's a lot of very popular dive and snorkel site, and then maybe octopus are staying out of that swim area for that reason.
Great. So another guest mentioned that den site concentrations in your study area appear to be very dense. Is that an anomaly of this being a 3-year composite, or are there really that many dens at any given time in your study area?
There are, so a little bit of both. It may look like more because you're looking at 3 years worth of data. However, when I did look at this, so I would partition this out and look at it year by year to make sure that I saw the same trends of them clustering in those areas. And we still do have high densities, when you separate those years. Out, and usually those high densities are seasonal, as I mentioned, during the spring and summer times. And again, I've counted, 5 or more octopuses on a single dive during the springtime. So density definitely increases. And
that's a good tie into our next question. Do you have any idea of what causes the seasonal population variations that you observe? And if it is migration, where are they migrating to?
Yeah, so more research needs to be done, but I definitely think that this, it's a nursery and a recruitment area. Again, this site is very close to the Palm Beach Inlet, and these two species, when they hatch, they're planktonic, but they're called paralarvae. So when they hatch, they float up into the water column, and they live in the water column for a little bit before they become a benthic animal. So they're able to, their distribution they can cover when they're floating is great. So they could come into the Lake Worth Lagoon inside the inlet, settle as juveniles. And juveniles, when you're an octopus, and most juveniles, when you have a lot of predators. So the idea is that these juveniles come into the shallow, warm lagoon where there's a lot of prey. They can eat, they can hide from predators, and they can grow super fast. Once they are ready, once they are mature, the idea is that they migrate out of the lagoon to deeper waters to slow their metabolism. It's shown that adult octopuses will do that. However, I haven't been able to do all of that. And it would be great to look at their migration if they are, coming and going out of the Lake Worth Lagoon, more camera traps or OMGs set up, ROVs to set up, or also octopus tagging.
I guess I was going to ask us that question. Are there techniques to tag octopuses?
There are. There's been some where it has to be, when they mark them like a little stamp or barcode, but then you have to do catch and release. That can be tricky to find an octopus again. Larger species like the giant Pacific octopus, acoustic tag was actually developed and put on there so they could track the migration patterns of the giant Pacific octopus. However, those tags are becoming much smaller, and I think it is possible to tag these smaller species. So
very cool.
Hopefully a future research project.
That'd be awesome. We've actually had quite a few people wondering whether you had an opinion on the Netflix documentary, My Octopus Teacher. This is a kind of a reoccurring question worded a few different ways, but it's definitely popped up a number of times tonight.
Yeah. I think I'd have to watch it again. I feel like I'm gonna get shunned because I haven't watched it fully. I started it late at night and I fell asleep. So I've seen most of it, and I know what happens. But I do appreciate the documentary, of course, showcasing the octopus as this amazing animal. Of course, I am the cheerleader for octopuses and how important they are to marine environments. And the cinematography in that film is amazing. And so it's nice to see them showcase that.
All right. So this is a really interesting question, I think. Is there a way to use fecal analysis to assess octopus diet?
Yes. yes, you can. I don't know how detailed you can get, if you can get down to species level like I did with the prey samples. It's possible. It's a little tricky. I like to describe octopus poop as silly string. It's like pink silly string. And so if you saw in the photos and videos that I've showed, I refer to the orange funnel or the siphon of the octopus. And so the siphon is responsible for pushing out water for jet propulsion. That's where the ink comes out of to scare predators. And then also that's where the silly string poop comes out of. And so as you can imagine, the octopus pooping and then it just kind of spinning and going in the water column, you got to be ready to go and to grab that fecal sample. So it's possible. I'm always up for a challenge. So I could do it. Yeah.
All right. What range do individual octopuses use around their den site? And does that vary between the two species you study?
So range for living range or
I'm reading this
foraging?
maybe as like foraging range, or like how, when they leave the burrow, how far away from that burrow do they?
Yes, that's another great question. And I think that everyone is reading my mind of the future research projects I want to do. I have followed them on foraging events, and they can be, 15 minutes up to 2 to 3 hours. So they can cover some ground. And we know that they have, spatial learning and memory and they use landmarks of where they go, because they can be in one area. If there is a predator around, they don't trace back to where they came from. They just beeline straight back to their den. So they know their surrounding environments, because they definitely have to. However, I have not marked out with, a GPS of the actual distance, but they do travel quite a bit. And I would love to, put on the GPS map and follow them to also see not only how far they go, but also that foraging range, if that overlaps with species. And I think it probably would.
Do you have any thoughts on the notion that octopuses are sentient species? I think this was recently an issue in Europe, right? Where they have labeled them a sentient species in terms of being a food item, and they have the feelings of, higher animals. Do you have any thoughts on that?
Of pain?
Yeah.
Yes, there has been, it's definitely an ongoing research topic. There has been a recent study that was published about if, they do feel pain. And I think it's important to understand reflex, a reflex that we have versus pain or reflex. If if we touch something and it's hot and you pull back and it's a reflex, versus pain has a behavior associated that you learn. Like if you see that red plate and you're like, oh, I know that's associated with pain, I'm not going to touch that. So just making sure that we understand reflex versus pain, and pain being associated with a behavior. That being said, a recent study was just done to show that octopuses, yes, they have the nociceptors feeling the pain, and a nice set of experiments was done with that to show, behaviors associated with pain, stating that yes, they can feel pain. And so that's been, a long talking, discussion between scientists, and of course, more research and experiments need to be done. But I think that we are taking an appropriate approach. We are scientists and we need data to back, what it shows before we make any abrupt changes. However, we are going into, into the future with, I guess, with committees at not only, universities and organizations, that they just don't let us do anything with animals. There's a lot of permits and protocols that we have to go through. And like us and the committees, we stay up to date on the latest, animal care and behavior. So taking these studies, implementing that, if they do feel pain, how we're going to move forward with what's appropriate, and how to do appropriate octopus behavior experiments.
All right, we've got a biomechanics question. How does the octopus's drill actually work? Does it somehow spin like a Dremel tool, or is it stabbing like a needle?
That is a great question. And I don't know the nitty gritty biomechanics behind it, but whenever I don't know a question, I always find myself going down the research rabbit hole and finding an answer for you guys, since it is, hard to see if an octopus is, if it's drilling, you can't actually observe like what it's doing. But of course there are, dissections and describing the drilling of the salivary papillae. So definitely I'll look into, what that description is. When I've seen pictures and everything, it actually looks like a little drill, if that is spinning or going back and forth. It's a great question.
We have a couple minutes left. I'll try to get through as many of these as I can. This is actually a tie-in to the previous question, but from a different viewer. They believe that certain gastropods will also drill a hole in their prey. Mm-hmm. So how do you determine whether the holes that you're seeing in these prey items come from an octopus or from a gastropod?
Yeah, that's a great question. And so depending on the animal, the size of the drill hole and the location of the drill hole help us determine if it was an octopus or it was a gastropod or another animal that drills this hole. Also, since I'm taking these from octopus dens and making sure they're newly discarded prey, there's not algal growth or anything on them that I know they've just been sitting there, I'm pretty confident that these were from octopuses.
All right. So one of our viewers tonight is tuning in from the west coast of Florida, and they see a lot of crab traps, both inshore for blue crabs and offshore for stone crabs. They're wondering whether there's any indication that octopuses raid those crab traps for an easy snack.
Oh yeah, I hear about that from quite a few people having octopuses in their crab traps. And I was actually in contact with a couple of them because I was trying to figure out how I could do more sampling for my octopus swabbing project, because there are fishermen that pull up the trap and there's an octopus in there. And I was going to say, contact me if there is, because I'm going to run down while you have the octopus and swab it before you release it. So, yep.
That's one way to collect data.
Yeah. All options. I'm trying.
Yep. So in your opinion, what are the main threats to the existence of octopuses? Pretty important question.
Yeah, definitely. Main threats to octopuses. I'd say habitat degradation, which is, a main threat for many animals. So habitat degradation for them, but also for their food source. Like I mentioned, of course, climate change and ocean acidification. That also has to do with anthropogenic threats. So not just thinking about warming water temperatures and acidification, but also pollution, thinking of runoff waters and the contaminants and what's in there, possibly plastics, plastic additives, but also metals. This is, again, going back to the Lake Worth Lagoon and also Florida being very urbanized area, and what after we have a large rainy season, what's coming in the water, thinking about these hard metals that or trace metals that the octopus is coming in contact with and how they are coping with that as juveniles and adults, and if that is altering their behavior. And that can definitely alter their foraging behavior, and if they can't forage to feed, then they're not going to survive, and it kind of just keeps going up the food chain from there. So a lot of threats that a lot of other animals are facing too, the octopuses are facing as well.
All right, so looping that back to climate change and range expansion, is the Octopus insularis in Florida causing any harm that we know of besides typical competition? Do we have any info that it's out competing the other octopus species, or can they coexist? Do we even know enough to make that decision at this point?
We do not know enough to make that decision. This paper that, this information was published just a few months ago. We just, I just found out, I just discovered that this species is here in Florida. And now, it's kind of future research to see how abundant it is, where exactly it's located, what habitat it's taking up, and to go from there. Yeah, so I can't make any conclusions yet.
All right. Do the long-arm octopuses line their den with a substance to prevent the sand from collapsing?
That's a great question. I do not know if they do. However, I know that other sand-dwelling octopuses do, and it might be kind of like a mucus layer so it doesn't collapse or fall in. And that is another research project that I would love to do, is looking at, the tunnels that they create again, kind of looking at like ecosystem engineers, the tunnels that they create, if they're altering the substrates or any way, how they're keeping these burrows, because it's, you know, it's been observed a couple times that they will create a burrow and then make a little tunnel and then they'll pop out somewhere else. So how exactly do they do this? How deep are they? What all is going on? And so I don't know if anyone remembers the ant farms that were, you know, so big, but then you could see the tunnels that the ants would make. Kind of want to do that, but octopus style. So you can study the burrows of the octopus to see what exactly is going on.
Chelsea, you have so many great questions in queue here. I'm not going to get to all of them, but I'm going to make an executive decision to go maybe a minute or two after 8 to see if I can get a few more of these in. These are pretty easy ones. How long do octopuses live for?
Yeah, in general, octopuses are short-lived. The two species that I study live for about 1 to 2 years. A lot of species are 1 to 2 years. The largest octopus, the giant Pacific octopus, you know, over 200 pounds, large octopus only lives 5 years. So for a very large animal, that's really, really short.
Are octopuses able to make noises?
Able to make noises? I'm like, what kind of noises? I don't think I don't think so. Like, I haven't heard any. I'll listen next time. I'm sure that, like, I don't even know if like something with their sucker like coming off, but like I haven't heard any noises. No.
All right, here's one that may rub some people the wrong way. Some people refer to many as octopi, but is it correct to say octopuses?
Octopuses is correct. So it comes from the Greek, not the Latin, octopodes. And so when you, I guess, translate that into English, it takes the English plural ending of es. So plural octopuses is correct.
Do you have any crazy diving experiences with octopuses or any other animals in your research that you'd like to share with us?
Oh man, crazy experiences. It's always a good time on an octopus dive. I think, you know, some of my favorite dives is, you know, when they are curious and they come out, like on the video when that one just jumped on my camera. That's happened a few times. Or when I will leave my camera to the side and I'll be busy with another octopus swabbing, and another octopus will come over and we'll check out my camera. So I love their curiosity. They have some crazy behaviors that I didn't even get into that, you know, maybe I can just do a presentation about just octopus behaviors. I love their behaviors. I especially love the Atlantic longarm octopus. Just like I mentioned, it's crazy how this is a shallow water animal. You know, sometimes I see at Blue Heron in 3 feet of water, but we really don't know much about it. Besides my studies, I think there's maybe a handful of papers or under that have done research on this animal. So although like it's there, it's in shallow water, so we don't know much about it, and it does some really cool behaviors. So yeah, any crazy behaviors on my research dives are great. And it just makes me laugh just to show you how focused I am on a research dive. And of course, octopuses are benthic, living on the bottom. So my head is always down and I'm always working with the octopus. So there have been manatees that have swam right next to me and I don't even notice. My dive buddy will be trying to get my attention to be like, there's a huge manatee right next to you. And I'm completely oblivious because I'm with an octopus doing research. So I really like those stories as well.
Chelsea, do you have time for maybe 2 more questions? We actually still have about 150 people tuned in. Okay. At our peak tonight, we had around 240. So you still have a big audience. I'm not going to get to all these questions, but I see 2 more that I'd like to try. In some of your foraging videos, it looked like a fish was staying close to the octopus. Is this a behavior you see regularly? Was the fish waiting for the octopus to flush something out for the fish to eat?
Yes. Excellent question. And that brings up like a great topic that I'd also like to talk about. So Yes, it is common to have maybe one fish or multiple fish follow the octopus on foraging events, and they are correct. These animals are waiting for the octopus to flush something out of maybe a crevice or algae or a hole. So the fish pretty much is kind of like taking advantage or giving a free ride and grabbing, you know, a meal that maybe the octopus flushes out of a hole. Sometimes the fish will also actually nip at the octopus arms. And so I don't know if this nipping, if they actually sometimes get a piece of the octopus's arm, or they'll nip at it. And so I don't know if you've seen in the recent news or memes about octopuses, you know, not caring, they just punch fish because, you know, out of spite because they want to. And I have to be like, no, they have good reason for punching this fish. There's multiple fish following them that could possibly be blowing their cover, you know, from a predator, and they're trying to get a free meal and they're biting the octopus's arm. I'm like, I'd punch a fish too if I was an octopus. So I'm team octopus when it comes to that.
All right, and the final question for the night, this is a great tie-in to next week's lecture about nurse shark reproduction. Somebody wants to know, how do octopuses reproduce?
Yes, so octopuses, what we'll do, and that is also one way, usually the male and the female octopus look exactly the same. Usually there's no sexual dimorphism except for in certain species of octopus. So the blanket octopus has one of the most extreme sexual dimorphism there is, the female being like 6 feet long and the male being like walnut size. However, for the species that I study, you can't tell if it's male or female unless they are mating. And so usually what they'll do, using their chemotactile cues, they'll see that, oh, this is the same species, opposite sex. Octopuses are solitary animals, so when they find a mate, they go for it. And so the male octopus will extend, it's his third arm. It's usually his third right arm, called the hectocaudalus. So it's a hectocaudalized arm. And so the end of that arm is specialized. It has a special groove down it. And so that's to pass the packet of sperm. So they'll pass the packet of sperm from their gonad and their mantle all the way down that third arm in that groove. And that male octopus will stick his hectocaudalus arms, I call it the sexy arm, to the female inside her mantle where he deposits the packet of sperm. The female can then decide, she can actually hold on to it for a few months if she wants, to decide if she wants to accept his sperm to fertilize her eggs or not. And just like sea turtles, there can be multiple fathers for the eggs that the female hatches. So fertilization is internal. However, then the female octopus finds a nice safe spot where she lays her eggs and guards her eggs for the rest of her life.
Chelsea, I can't thank you enough for hanging out with us tonight. This is a really cool presentation. I learned so much. I know our guests did. I could tell by the number and quality of questions that we got that our audience absolutely loved your talk. And hopefully maybe a couple of years from now, you can give us a follow-up on your research and give us some updates.
Absolutely.
For all of you who are attending tonight, I really hope to see you for our final lecture a week from now. If you enjoyed Chelsea's presentation, I think you'll also enjoy Jeff's talk. Very similar in terms of heavily weighted with underwater videography and great photos, and also a similar type of story, a long-term study working with an organism, in Jeff's case, nurse shark reproduction. But either way, if you like scuba diving or if you like spending time in and around the water, I think you'll really enjoy next week's presentation, whether you tune in on Zoom or you join us live at the Coastal Center with our limited capacity audience. Chelsea, thank you again. Really, really cool presentation.
Thank you.
Thank you to all of our audience members. Good night, everyone.